Heart muscle is a type of involuntary striated muscle found in the wall and histological foundation of the heart, specifically the myocardium. Muscle cells. The cells that constitute the cardiac muscle, called cardiomyocytes or myocardiocytes, contain only one unique nucleus. Cardiac muscle cells, like a tissues in the body, rely on an ample blood supply to deliver oxygen and nutrients and to remove waste products such as carbon dioxide. Structure. Striation. Cardiac muscle exhibits cross striation formed by alternating segments of thick and thin protein filaments. T-tubules. The T-tubules in the cardiac muscle are larger and broader and run along the C-disc. Intercalate discs are complex adhering structures that connect single cardiac myocytes to an electrochemical syncytial.
Brought to you by the Phillips Learning Center. The full e-learning course on cardiac structure and function can be found at theonlinelearningcenter.com under the cardiology section of the course catalog. It is a large, hollow This course reviews an overview of the cardiovascular system and its components, various cardiovascular anatomical structures, conduction systems of the heart, and physiology of the sliding filament theory. The heart is located almost at the center of the thoracic cavity. It is a large, hollow muscular organ that pumps blood throughout the body. The heart contracts rhythmically to distribute blood to all parts of the body at an average rate of 72 BPM, beats per minute. Let's view the circulatory system. The circulatory system consists of red blood cells, white blood cells, and platelets. The heart lies between the pleurae of the lungs in the thoracic cavity called mediastinum, with enclosed pericardial membranes. The heart is composed of three layers: endocardium, myocardium, and epicardium. Let's view the anterior, right, left, posterior, and inferior borders of the heart. These are the different angles of the heart. This is the posterior view of the heart from outside. Here you can observe the ventricles, aorta, pulmonary arteries and veins, superior and inferior vena cava, posterior coronary artery, and coronary sinus. Here, the anterior aspect of the heart shows the aorta, superior vena cava, pulmonary trunk, and cardiac vessels. Let's take a closer look inside the heart. These are the four chambers. The upper chambers are known as the atria, and the bottom chambers are the ventricles. The atria are divided into the left and the right atria, and the ventricles are divided into the left and the right ventricles. The function of the atrium is to receive the blood while the ventricle pumps it out. These are the valves of the heart. They are opened in the direction of blood flow and closed in the opposite direction. Their function is to prevent backward flow of the blood to the heart. These are the atrioventricular valves and the semilunar valves. These are the main components of the cardiac conduction system, which cause the heart muscles to contract rhythmically. They are the SA node or sinoatrial node, atrioventricular node labeled as a V node, bundle of His, bundle branches, and Purkinje fibers. According to the physiology of the sliding filament theory, the movement of thick and thin filaments is relative to each other, Rn, and leads to the contraction and relaxation of the entire muscle.
Structure. Physiology. The myocardial tissue is excitable and has four fundamental properties: excitability, automatism, impulse conduction, and contractility.
The
sequence cell across the voltage-dependent L-type calcium channel. The calcium that enters the cell triggers calcium release from the ryanodine receptor through a process known as calcium-triggered calcium release. Calcium that is released from the sarcoplasmic reticulum binds to the actin-thin filaments, thereby initiating contraction of the sarcomeres. At the end of the contraction sequence, calcium comes off of the myofilaments and is actively transported back into the sarcoplasmic reticulum by the calcium pump in its membrane. Calcium is also transported out of the cell into the extracellular space by a membrane-bound pump sodium-calcium exchanger, which is not shown here. Cardiac contraction is brought about by interactions between the actin-thin filament, shown in yellow and purple, and the myosin cross bridges that project from the thick filament, shown in red. In relaxed cardiac muscle, the conformation of troponin and tropomyosin prevents actin from interacting with the myosin cross bridges. As a result, actin is unable to interact with the myosin cross bridges containing bound ATP. In activated cardiac muscle, calcium binds to troponin C, which then shifts troponin complexes and tropomyosin to an active conformation that enables actin to interact with the myosin cross bridges. Myosin ATPase on the myosin heads then hydrolyzes ATP, thus providing the requisite energy for the myosin cross bridge to interact with the thin filaments and pull the thin filaments towards the center of the sarcomere in a ratchet-like fashion. In this sequence of the normal heart, we have reviewed the process of excitation-contraction coupling. As will be discussed subsequently, this process of excitation-contraction coupling becomes dysregulated in cardiac hypertrophy and cardiac failure.